own neutrinos because they're sterile and I like the taste. Oh, I'm your host, Chris Bickham. Today the Los Alamos National Lab announced it had once again detected what may be a neutrino with mass, with no mass. Sterile. Los Alamos National Lab announced the detection, once again, of what may be a sterile neutrino. This neutrino does not interact nuclear weak force, only gravitationally. Propose this is a potential candidate for particles that may be serotonin. It's in your brain. It's in your gut. You'd be surprised the function it has throughout your body. SSRIs, for example, are a treatment for depression. It has been clinically accepted as having a positive effect on this receptor system. A test. Find a friend who's taken this type of medication and has experienced 0 negative side effects. It's okay. I'll wait. Psilocybin and lysergic acid diethylamide are serotonin-binding compounds, which possess some very interesting binding properties. With lysergamides, the receptor pretty much hugs them and holds them like an inappropriately long, okay, we're doing this kind of hug. Current drug scheduling is seen by many as no longer productive. Our need to treat cancers, diseases, mental illness is becoming so comprehensive that we must re-evaluate the outcome our policy is intending to produce. May 29th, in the Journal of Psychopharmacology, a study found men with some history of psychedelic drug use, specifically, negatively correlated with violence against a significant other. This is in congruence with other studies showing a significant difference between this class of serotonin-binding compounds and other drugs. The sample size was 1,266 people, aged 16 through 70. The average age was 22.78 years old. This population is normally distributed. That's a good thing. However, this study did utilize self-reporting, which is notoriously squared. The protocol is listed. Experiment subjects were prompted to disclose all use of psychedelic compounds, specifically psilocybin-containing mushrooms and lysergic acid. Following this, they were questioned on the other pertinent, hopefully non-violent parameters, questions pertaining to emotional regulation. The researchers believe that the increased capacity via emotional regulation may be a potential mechanism to describe the negative correlation between the compound's utilization and partner violence. This is not the only study to share these results. Consider the source, consider the experiment, consider the data. You may think I'm regardless, that's super fun science. Expansion in the realm of deoxyribonucleic acid pathways to proteindom is an ongoing and tumultuous journey. A study entitled Genome Atlas of the Human Plasma Proteome in Nature has produced a map, quote, 4-fold larger than previous models. This study focused on plasma proteins, unraveling the cis and trans functions in our DNA. Don't be a punk, they're just words. Cis are often referred to as the non-coding, but they're really not. They don't build shit, they sometimes tell other shit what to do, like your supervisor at work. Trans refers to the coding regions for proteins to be built. Trans is short for transcription. They're blueprints. Here are some figures from the study relating to gene expression during disease. Say, irritable bowel syndrome. The amount of data here is significant, and this episode will be 47 hours long if I mention everything that is relevant. Trigger warning. For those of you who get nervous when hearing words like biology, evolution, genetics, you can't argue with this kind of data set. It's replicable. It exists in all of us. The proof is
in the motherfucking pudding.
You're part Neanderthal. Actually, some of you, Neanderthal. We're all primates. The code is too long, and thousands of coincidences don't simultaneously occur in a row over and over. You ignore the science, it can actually be mathematically proven in this case. You're 100% being dumb as fuck. In the bioengineering field, I remembered my first course working on my freshman design project. I read a Stanford paper that left me fucking flabbergasted. They devised a polymer skin covering equipped with cleverly designed pressure sensors. Squishy pyramid polymer regions were filled with a distribution of carbon nanotubes. Under physical deformation, their properties changed. These were utilizing the system to send signals from 0 to 200 Hz as sensory data out from the skin. I would also say respect to Brown University, or whoever it was that published the paper that I am now unable to find. Paper over the mapping of gripping motions in digits. They implanted microelectrode arrays in the motor premotor cortex of monkeys. It was from this I discerned, damn, look at the neural firing pattern. It actually looks like stick figures of hands. Is it possible the brain actually approximates 3D models just like we conceptualize things before sending signals out? I don't know yet, but that thought consumed me for a while after reading that paper. Ohio State produced an artificial nerve bypass on the arm of a wounded veteran. A lot of others I didn't mention as well. As the kids say, my bad.
Damn.
A lot of work from a lot of universities went into the development of these technologies. All of this shit under discussion has transpired over the past decade. Actually, more like the past 5 to 6 years. To give an overview, we confirmed, mapped, and successfully integrated signals to and from the premotor, motor, and somatosensory cortices in the brain. We currently are limited by the differences in the circuits we use to sample, process, and transmit information in native neural tissue. Microelectrode arrays and approaches to actually sampling the neural output is a large component of this. EEG proponents have had some interesting successes using AI to interpret signals, but I view this as a flawed approach, as the sheer signal to fucked ratio involved in detecting complex three-dimensional network neuronal firing residual propagation through so much fucking tissue. This article zeroes in on a more specific dynamic. It was developed a synthetic afferent nerve. Biological systems operate in hierarchical parallel networks across the body. This implementation performs a task that has been achieved. However, the significance lies in how the task was performed. The system feeds the voltage potential changes from pressure sensors. This feeds to, it's an odd-numbered arrangement of CMOS inverters, forming a ring oscillator. The oscillator turns the voltage changes into voltage spikes in a frequency range of 0 to 100 Hz. Finally, the dessert awesome sauce lies in the design of a synaptic transistor. Uses an ion gel as a dielectric in a bioanalog of a field effect transformer. This dynamic, using a viscous ion solution, is much closer to how our neurons operate with neurotransmitter gradients. Depending on the amount of binding, the rate at which a neuron will respond will vary. Further, the system can integrate multiple sets of inputs and was ultimately used to detect Braille characters. It was also used in a cyborg cockroach system, but that's it. Today.